Hey guys, so I decided to make a quick biology video, final, for you guys to study, since I know it's like a class we have struggled a little bit with, but we'll do good on it. So I just decided to make a quick review on the things we have learned. So I'm going to use the study guide we were given. If you have any more questions, please go ahead and leave them below, and I'll answer them. So the first question is, so what are the stages of the cell cycle? So in the cell cycle, we have 4 different phases. The first one is called G1, the second one is called the S phase, the third one is called the G2, and the fourth one is called the M phase. So G1, S, and G2 are just kind of interphase. So don't get this confused with like interphase, prophase, metaphase, and all that. Those are mitosis. This is just a cell cycle overall. Within the cell cycle, you have mitosis and meiosis are kind of just coming after that. But just don't get them confused. They're two different things. Next question: what happens during interphase? So during interphase, DNA is replicated and it prepares for cell division. Next question: during which phase does DNA replication occur? Again, same thing, just like rephrased. Again, it happens during interphase. So now we're gonna go through mitosis, each of the steps. Again, okay. So during interphase, DNA is uncoiled, it replicates and prepares to divide. During prophase, DNA coils up and sister chromatids pair up. The nucleus breaks down and spindle begins to form. During metaphase, the chromosomes line up, spindle gets ready. During anaphase, the spindle attaches and pulls chromosomes apart toward opposite ends. And during telophase, the nucleus starts to reappear. And you might see cytokinesis. Cytokinesis is just like the final outcome when you have 2 cells completely separated. That's just kind of what it is. Please, I'm telling you, take a look at the cells, because you may have to identify them in actual paper. So just know how all of them differentiate. Okay, next question. What are the stages of mitosis in order? So again, I went through this. The first stage is called interphase, the second is called prophase, the third one is called metaphase, and the fourth one is called anaphase, and the last one is called telophase or cytokinesis. I'm struggling with this, but I will get it right now. Sun. So what are 3 differences between mitosis and meiosis? So mitosis, we have body cells, and meiosis, we have sex cells. Mitosis, we have 1 division. Meiosis, we have 2 divisions. Mitosis, we have 2 daughter cells as an outcome, and meiosis, we have 4 daughter cells as an outcome. The chromatin, and then in mitosis, there's no homologous pairing, while as meiosis, we have, sorry about that, we have homologous pairing, so that's like a huge difference. But if you're confused with what meiosis and mitosis is, so again, our body goes through cell division. Again, cell division, if you don't know what that is, just like the process of cells dividing. But we have 2 different processes simply because of the fact that we have a lot of our cells in our body. We have 2 types, which are somatic cells, also known as body cells, and sex cells. So the body must go through each one of these processes in order to fully carry out the division process. So just don't get confused. It's just like we have 2 different kinds of cells, so the process must go through undergo 2 processes. Next we have, what are 3 similarities between mitosis and meiosis? So spindle moves chromosomes to the middle, the nuclear envelope breaks down, the chromatin condenses and attaches to spindle. That's just 3 differences that happen in both of them. Okay, again, what kind of cells undergo mitosis? Body cells or somatic cells? You may see it on the exam as somatic. It means exactly the same thing. Don't get confused. Then we have what kind of cells undergo meiosis, and these are sex cells, such as sperm cells and egg cells. So what is the result of mitosis? So again, the result is you end up with 2 identical daughter cells. And then what is the result of meiosis? So you end up with 4 different haploid cells. Again, these 2 things are going to come about later: diploid and haploid. Diploid is just like the full genetic, meaning 46 chromosomes, and haploid is just like half of the genetic material, aka 23. Next question. So what happens in DNA replication? DNA is replicated. That is kind of self-explanatory. I don't think you should know that, but just know that might appear in the exam too. So what are the 3 steps of DNA replication? So first step is DNA molecule uncoils at any point of the DNA. Second step, new nucleotides are added to the template strand. So remember, we have two different strands. We went through this, I don't think you remember it, but we have the template strand, which is the original, or the parent cell. We have the daughter strand, which is the new strand, which has been coded. Those are the two. For this second step of DNA replication, we're talking about the template strand, which is the original strand. And the last step is we have two identical copies of the DNA molecule that are produced. Next question is, okay, when does a genetic mutation occur? So a genetic mutation occurs when they're present because of the parent's egg or sperm cells. That's it. Don't get it confused. If you ever see the word genetic, it doesn't refer to anything that can be caused once you're maybe environmental factors. When you hear genetic, it means that you can't see this, it's not affected by the environment you're in or anything like diseases. It's all in your genes from your parents and your own genetic material. Next question, when does a chromosomal mutation occur? Now in this case, we're talking about chromosomal mutations. Still, these are mutations, except these are not happening as a result of genetic whatever happened in the genes, I'm not sure. This happened as a result of environmental factors, such as ultraviolet radiation, or if a mistake is made as the DNA copies itself. And they cannot be passed down to the next generation, just know that. So, while genetic mutations can be passed down, chromosomal mutations cannot be passed down. What are the base pairing rules for DNA? So, again, we went through this. We have 4 different nitrogenous bases. So we have adenine, which binds with imine, and vice versa. Then we have cytosine, which binds with guanine, and vice versa. Remember that for RNA, this changed a little. I'm gonna get into that later. Okay. So why is base pairing important in DNA replication? It is important because a base can only pair with a definite pair and not just with any base. Therefore, this ensures high fidelity in replication. So just know that you can pair anything with whatever you want. It has to be exactly how it is supposed to be. Otherwise, it's just not going to work, and you'll have a very bad cell. You might even get some mutation, as we talked about. So next we have, what are the characteristics of RNA? So for RNA, we have, they are single-stranded. They contain ribose instead of deoxyribose. And it contains uracil instead of thymine. Next we have, what are 2 differences between DNA and RNA? So I actually have 3 differences because I like to be extra, but please just know 2. Though it would be maybe convenient to know 3, but DNA is double-stranded, RNA is not. Sugar in DNA is called deoxyribose, and in RNA is called ribose. The bases in DNA are adenine, guanine, cytosine, and thymine. The bases in RNA are adenine, guanine, cytosine, and uracil. So just know that uracil isn't kind of the one that's not present in DNA, and that's kind of one of the main differences. But yeah. So next question is, what is the relationship between DNA, genes, and alleles? So DNA is a double-stranded nucleic acid that contains the genetic information for the cell growth. A gene is a segment of DNA on the chromosome that is coded for a particular trait. Note, for a particular trait. Then we have an allele, which determines the specificity of the trait, found in each gene. So each gene has 2 alleles. These genes are found in the big ladder you see in DNA. So alleles, again, don't confuse this. Alleles means like a specific thing, like red hair, brown eyes, just things that's specified for a specific color trait that you will visibly see. Again, a trait could just be like hair or eye color. So I'm not specifying what that is, as opposed to when I'm in an allele, I am.